Hello and welcome back to yet another video. In this video I'll talk about enzymes, and in the main course I'll talk about how they work, and in that I'll explain the lock and key theory of enzyme activity and the induced fit theory. So let's get into it. Okay, now enzymes are protein in nature and they catalyze metabolic processes. Now I'm sure we know, actually most of us know, that a catalyst speeds up the rate of a chemical reaction. So enzymes are basically biological catalysts because they are protein in nature, and they are made by our living cells in a process called protein synthesis. So just like any other catalyst, enzymes speed up the rate of metabolic processes and remain unchanged at the end of the reaction. And this allows them to be used repeatedly throughout these metabolic reactions. Now, almost every metabolic reaction, whether it involves breaking down molecules, these are catabolic reactions, or building up molecules, all these reactions require enzymes. Now, an important thing to note is that enzymes do not make reactions occur. Rather, they simply speed up the rate of these reactions because these metabolic reactions will take place too slowly to sustain life without enzymes. Now, for any typical reaction to occur, any typical metabolic reaction to occur, there has to be a little energy put in to kickstart the reaction, and that is what we call the activation energy. It's that energy needed for a reaction to occur, and this activation energy is usually very high when a metabolic reaction is taking place without an enzyme. So enzymes, by functioning as a catalyst, rather a biological catalyst, serve to reduce the activation energy required for a chemical reaction to take place. So the activation energy is much lower when an enzyme is involved in a metabolic reaction. Now, the molecules on which enzymes work on are called substrates, and these can be proteins, carbohydrates, and so many others. Now, an enzyme combines with its substrate to form a short-lived enzyme substrate complex. Now, once this reaction occurs, the complex breaks down, the substrate rather breaks down into products, and the enzyme remains unchanged and is free to interact again with more substrates. Okay, now I told you enzymes are protein in nature. Now, enzymes also have a non-protein component, and this non-protein component of an enzyme are called cofactors. So cofactors make up the non-protein component of an enzyme, and this helps the enzyme to function more efficiently. Now, cofactors include inorganic ions such as sodium ions and chloride ions, and these activate the enzyme, so they're required. They basically make the enzyme active. Then next are the prosthetic groups and coenzymes, and these assist in activation and catalytic activity of the enzyme. Now these are usually ignored, and I'll also kind of ignore them, so you can do more research on that. Now anyway, an enzyme possesses a small functional region where catalytic activity takes place, and this is called the active site. And as you can see, it has a specific shape, so this is what makes the enzyme specific to the substrate they bind with. And with that said, we can now move on to how enzymes work. And there are two main theories that explain how enzymes work. There is the first is the lock and key theory, and the more accepted induced fit theory. And I'll start with the first and original theory, the lock and key theory. Now, from the name alone, the enzyme and the substrate work in the same way. A key operates a lock, and each key has a specific shape to fit and operate only one lock, though some people have mastered how to change that narrative. Anyway, the substrate here is let's imagine it's our key, and the enzyme is the lock. Now this substrate will only fit the active site of an enzyme whose shape is complementary to the key. So according to this, the enzymes are therefore specific in the reaction they catalyze. So the substrate, according to this theory, will bind with the active site of the enzyme whose shape is complementary to its own shape, and this will form an enzyme substrate complex, which eventually the substrate breaks down, forming products, and this leaves the enzyme free to interact with more substrates. And that is what you need to know on the lock and key hypothesis. Very simple to understand. However, the lock and key theory does not give a totally satisfying explanation of enzyme action. And the induced fit theory more than compensates for that. Now the induced fit theory is a modified version of the lock and key theory. Now according to the induced fit theory, the active site of the enzyme is more flexible than previously described in the lock and key hypothesis. So this basically means that the active site of the enzyme is flexible and therefore molds itself into a precise shape complementary to the shape of the substrate it's interacting with. So the shape of the active site is influenced by the shape of the substrate, and this enables the enzyme to perform its catalytic function more efficiently. And therefore, the substrate is able to bind with the enzyme, forming a short-lived enzyme substrate complex, and this the substrate later breaks down into products, and this leaves the enzyme, and the enzyme returns to its original shape, ready to interact with another substrate. And that is the induced fit theory in a flash. Now, if you enjoyed watching this video, even if you did not, but if you have watched to this far, please like the video, subscribe to my channel, and I'll see you in my next video. Thank you for watching.